In this video we will discuss about genes that are involved in the trafficking or recycling. So trafficking and recycling means the recycling of the proteins either to the plasma membrane or to the trans-Golgi network or between endosome and lysosomes. These are the two categories that are discussed in this figure. So first they have provided the result of this retromer and retriever. The retromer means the recycling of the integrins to the trans-Golgi network, and the retriever means recycling of the integrins to the plasma membrane. And simultaneously they also checked some results for this hops and covert, and these kind of genes that helps in the protein trafficking between endosome and lysosomes. And what they are able to find out is that the maximum down regulation occurs in the VPS29, and it should affect the integrin levels on the cell surface. In order to validate that hypothesis, they develop a stable cell line of this VPS29, and for negative control they use this ITGV2, which will decrease the signal significantly for the CD11B or CD18 or these integrins expressions. And for the positive control they use SNX17, which is known to play a role towards the integrins expressions to the plasma membrane. And what they are able to see compared to the control, they see a decrease in all of them, maximum decrease in the negative control, and thereafter it comes the SNX17 and VPS29 signals also low compared to the this reference control, where none of these catenaries are being knocked out. So this indicates that the VPS29 also has a role towards this protein trafficking, basically in the retriever regions, that is traffic helps in the recycling of the proteins to the plasma membrane. And from this 3D amyloid motion they are also able to find out some of the genes that are involved in this protein and trafficking. Here they checked it for the alpha subunit and beta subunit of different genes, and they are able to see for the ITGA1 genes, which is highly downregulated, and this ITGA1 genes is known to affect the integrins that plays in role towards the interactions with the collagen. So they should expect a decrease in the migrations or persistence when this ITGA1 gene is been knocked down. So they develop a stable cell lines of ITGB2 genes, and for the control they use ITGA1 genes. And when they check the average speed, they are able to see that there is no difference in the average speed in either of this knockdown compared to the control. But when they checked it for the persistence, they are able to see that the persistence are significantly low in the case of this ITGA1 gene knockdown compared to the control, and also for this ITGB2 genes. So this in together indicates that the results that they are seeing are results whatever they have seen in during the CRISPR screening seems to be correct, and the results, the validation that they have done using the persistence, average persistence, and the flow cytometry analysis gives more authenticity to the CRISPR screening results. So in this case, this SG control is the reference control that they have used, and this ITGB2 we can consider it as a control to see whether this effect that they have seen in the ITGA1 knockdown is unique to the ITGA1 or it is common in any knockdown cell lines. That is why they use this ITGB2. And similarly for in the panel B, when they check it for the retriever, here they have used this SNX17, which is already known in the literature, from the previous literatures, that it will help in the recycling of the integrins to the plasma membrane. So from this and also from the negative control, they are able to make a point that the experimental protocol is same, and it's not, the results are not the artifact of their experiments. how the experiments are carried out. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.